Hello, this is Mr. Bradley. Today I'm going to be teaching my Biology 1 lesson on the movement across the membrane. So this will cover chapter 5-2 and 5-3 in our textbook. So the first thing I want to talk about is the actual structure of the membrane. So this is a zoomed in picture of the cell membrane. So like let's say if we're looking at a cell here, this would be a picture of just a tiny little portion of the cell. And what you see here is that the main part of the cell is a bunch of these little things here called phospholipids. So letter A here is referring to a phospholipid. And a phospholipid makes up what's called the fluid portion of the membrane. So if you imagine that, if you've seen like a soap bubble, and you see how the soap bubble looks like it's kind of liquidy on the surface. You can kind of see the fluid moving around in it. The soap bubble molecule is very similar in composition to a phospholipid. And what makes it special is that it has two regions. It has a hydrophilic region known as the phosphate head, and that's labeled A1 right here in colored blue. And then it has a fatty acid tail, and it actually has two fatty acid tails coming off of it that make it hydrophobic. And hydrophilic means water loving, and hydrophobic means water fearing. So as a result, it forms this, what's called the bilayer, the lipid bilayer. And the lipid bilayer forms so that water is facing both this end here, and this is going to be what's referred to as the extracellular environment. And then there's going to be water here, and this is called the intercellular or the cytoplasm. And as a result of the water being on both sides, it forms this barrier that is a selectively permeable barrier. And what makes it selectively permeable is because lipids are going to be mostly composed of these fatty acid tails, it's going to prevent things that don't interact well with fats from passing through. So there's some other things that are going to be seen here. One of the big things that you see here is the thing labeled D, and that is a protein channel. And we've talked about protein channels earlier as one of the functions that proteins do, one of the main functions. And then this is going to allow larger materials to move through the cell membrane. They make a pore. Some other things that proteins do is they act as receptors. So the thing labeled B is a receptor. And oftentimes a receptor will have a region on it that's made up of polysaccharides or carbohydrates. And these are going to be attached to the phospholipids or to a protein to allow them to be it helps them interact with other materials, or it can also help them attach to other cells. So like these carbohydrates here, they might interact with other carbohydrates of another cell and kind of make it like Velcro where the cells will stick together. Finally, the other thing that you see here are cholesterol. So letter F here is a cholesterol molecule, and cholesterol will interact with the cells, and basically what it does is it helps to solidify the cell. It's a dense fat, and as it says there, what it basically is going to do is going to anchor this thing here or this thing here in place so it doesn't move around. Because again, this whole region, even though it looks like it's solid, is actually a liquid, and as that liquid moves around, all these things could move around. And as a result of these things being able to travel around like they're floating, it's actually referred to as a fluid mosaic, meaning that there are many proteins floating in the membrane and their locations in the fluid change constantly. So that's the structure. Make sure you color this in for your notes so that you understand what each of these different parts are. And it's definitely crucial that you understand the terms hydrophilic and hydrophobic and how they relate to the phospholipid. So what does the cell need to do? So why is it important to learn about the cell membrane and membrane transport? Well, it's because the cell needs to be able to do all these things to maintain homeostasis. Remember, much of what you do that you consider being alive, breathing, eating, using the bathroom, are all related to maintaining homeostasis of your cells. So eating, you're going to get things like glucose, amino acids, nucleotides, fatty acids, and water. And you're going to do those things to get energy and to build things that you need to grow. So glucose is needed for energy in the process of respiration. Amino acids are used as building blocks of proteins, and you need those to build cytoskeleton, which is what's going to be the main component of your cells and your muscles. You're going to need them for enzymes, which you said help carry out chemical reactions, and membrane proteins, which act as channels and receptors, like we just showed. Then you have nucleotides. Nucleotides are going to be the building blocks of nucleic acids, and they're going to make up chromatin and ribosomes. And then you have fatty acids, which are going to be the building blocks for lipids. So key thing is that all this stuff comes from materials that you take in from your environment. So it comes from food and air and water. So you get this from breathing, and you get this stuff here from eating and drinking. So you eat and drink and breathe, and that oxygen is going to go into your lungs, and your food is going to go into your stomach and small intestines. And eventually, in both cases, they're going to get absorbed into your bloodstream. And the job of your bloodstream is to transmit all this stuff to your cells. Well, it needs to get from your blood into your cells. And that's what all these processes that we're going to learn about are involved in doing. So because the cell membrane is made up of lipids, which are non-polar, and the materials are dissolved in fluid outside of the cell are mostly made up of, which is mostly made up of water, is polar, these materials won't be able to move through the membrane. So as you see here, really small things like carbon dioxide, nitrogen, and oxygen, they're permeable. They can pass through that lipid bilayer. Ethanol is another thing that can pass directly through the lipid bilayer. Ethanol, by the way, is like what's in beer and wine and liquor. Water is slightly permeable, as is a material called urea, slightly permeable. Urea is going to be a main waste product. Things like glucose and ions, which I didn't quite mention those, but those are those electrolytes that you might need to get. Those ions are going to be important for various functions, generally cell-to-cell communication and also energy harnessing. But then your other building block materials, your nucleic acids, amino acids, those things are not going to be permeable. So all of these things here you see that would just not move through. So as a result, they're going to have to be able to pass through membrane proteins. So we're going to talk about two different types of transport. First is called passive transport. Passive transport is where the cell doesn't need to expend any additional energy to get materials to move in or out of the cell. And this type of movement into and out of the cell, as it says, it doesn't require any additional energy because of the law of diffusion. The law of diffusion means materials will move from an area of high concentration to an area of low concentration. So the easiest way to think of the law of diffusion, if I spray like cologne, like Axe body spray in one corner of the room, eventually those smell molecules will dissipate throughout the room until everyone can smell, and then eventually they'll kind of dissipate out even further till you really don't smell them anymore. So the other way to talk about the law of diffusion is to say that materials are moving down a concentration gradient. There's two types of passive transport. One is just called simple diffusion, and this is the movement of very small materials directly through the lipid bilayer. So if you look back at the diagram on the previous page, this one right here, the things that are going to be able to move directly through ethanol, carbon dioxide, nitrogen, and oxygen, those would be the things that'll move through by simple diffusion. Now, ethanol is not something that we need to survive, but carbon dioxide and oxygen are both things that cells need to survive. So in general, we talk about those things. Water can sort of move through. Nitrogen is a major component of the air, but we don't use it. But other organisms do use nitrogen, so its movement is important for living things. So why do you think it's a good idea for the cells to be able to move these two things so easily? Well, you breathe in oxygen and you're breathing out CO2. So your body is constantly using those, so you need to get into the cells easily. Like if you run out of oxygen, your body doesn't survive very long. Like maybe for just a little bit of time, you can survive without oxygen. The second type of passive transport is called facilitated diffusion or facilitated passive transport. It's how large and charged materials pass through the protein channel. So if you look at the diagram, the things that are going to not be able to pass through that you need, like glucose, ions, amino acids, ATP, those are going to be the things that are going to have to travel through a protein channel. So glucose, water, there are some protein channels that we've learned that move water called aquaporin. Electrolytes, they're charged only, so they can't pass through. Amino acids and nucleotides. So all those things have to travel through a protein channel. And it should be noted that there's protein channels not just on the outer cell membrane, but even to get into certain organelles, like the mitochondria has lots of protein channels as well. Active transport is movement through a cell membrane where it uses additional energy. So we talked about passive transport. This is active transport. So this diagram here is showing, here is simple diffusion type of passive transport going from high to low. This is facilitated diffusion going from high to low, but it's going through a protein channel. But if you look in this one, it's going from low to high. So active transport, because you're moving things against their concentration gradient, they're going to require extra energy, which is usually going to come from ATP. We'll talk about why the cell would do this, but I have this little diagram over here just to illustrate what we're talking about here. So a high concentration is like this. This is a low concentration. This, you're going up a gradient. This is going down a gradient. So up or against the gradient is active transport. Down with the gradient is passive transport. So diffusion, how does it actually work? Well, one of the things that you need to realize is that these materials like oxygen and things like this, they're not actually, there's nothing like moving them through. They are moving on their own. All materials are moving. So all the molecules that make up your body, they're in motion. You just can't tell because they're so tiny that you would never understand. But this is why things like particles from the sun, like gamma rays, can travel through you, because we are a lot of open space, and because we're made up of molecules. So those molecules are moving, so it like opens up space, closes space, opens space, closes space. The more gaseous something is, the more that they're going to be moving. The more solid it is, the less they're going to be moving. So like on the table, the wood molecules aren't moving as much as, say, the air is outside the table. When new molecules are added to an existing substance, natural movements will cause them to bounce off each other until they're evenly spread out. So diffusion is a random movement of molecules from regions of greater to lesser concentration. So gases diffusing in another gas, that happens relatively quickly. liquids diffusing in another liquid, that happens a little bit slower. Solids diffusing through a solid, that takes long time. The best you can kind of see with that is windows that get kind of old, so they're solid, but over time you can see the window of really old windows will kind of move down and areas will get thinner, and that basically you can kind of see like the windows look wavy. So new windows don't do that as much because they are what are called tempered glass. They basically put stuff in it to kind of prevent the glass from diffusing. So anyway, back to how this relates to the cell. If a membrane separates two fluids, like the inside of and the outside of the cell, the materials in the fluid could still diffuse if they're able to pass through the membrane. So oxygen can do this. So what you're going to illustrate here is you're going to draw these dots here representing oxygen in the blood. And then the oxygen is going to bounce until it gets into here. Now, again, it's not like there is an open space here. This is all going to be cells, and this is going to be the blood carrying oxygen. And usually, again, fresh oxygen is going to be moving past this all the time. So what's going to happen is this will always diffuse in. So this movement of oxygen continues until equal numbers on both sides. But since you're always getting new blood with new oxygen to the cells, there's always going to be a higher concentration outside the cell than inside. So there's always going to be movement of oxygen in, unless the heart stops beating or you stop breathing, and then that will cut off your supply of oxygen and you won't have movement into your cells. So there are things that are going to speed up diffusion. So one of the things you can do is increase concentration gradient. So higher concentrations means you have more bouncing off of each other and more likely to pass through the membrane, so faster diffusion. So one of the ways that we do that to get a higher concentration of, say, oxygen is you breathe faster. So when you're not getting any breathing, then you have no gradient. So there's going to be equal amounts. If there's a small gradient, you're going to get some movement of oxygen into the cell, but it won't be as great. But if you can get a lot of oxygen there, you're going to have a faster movement in to the cell because there's going to be a stronger push for diffusion to occur. The other thing that you can do is raise temperature. So part of the reason why our body temperature is as hot as it is because it helps the chemical reactions that are in our body work better. It helps us have the metabolism that we have. So hotter temperatures are going to make molecules move faster. Think of this, a good way to illustrate this is with tea. If you have a tea bag, you can do an easy experiment at home. Get 2 tea bags, put cold water in one and put hot water in the other, and put the 2 tea bags in and see how long it would take the tea to diffuse out of the tea bag in the cold versus the hot. The hot, it should diffuse faster. You could also do it with food coloring. You can have cold water and hot water. Add cold water to the food, cold water food coloring, or cold water to the food coloring to the cold water, food coloring to the hot water, and the hot water one will diffuse faster. The other thing that happens is cells will get different shapes, and cells that are adapted for allowing for diffusion, like the ones that are found in your lungs and in your small intestines where a lot of diffusion has to take place, they'll have special shapes that increase the surface area. So a cell with a small surface area will just have like a circle. So not as much diffusion happens in this one as the one that has lots of projections like that. So think of it as like, this would be like a tissue and this would be like a terry cloth towel. What's going to absorb more water quickly? This one won't absorb water as quickly as the terry cloth towel. So again, make sure you draw these illustrations. So note here, this says day 2. So this would be the next part of our lesson on osmosis. So if you wanted to take a break, this would be a good part to take a break on and then come back and finish this at this point. So osmosis is the diffusion of free water through a membrane. In animals, the net movement of water will be from the side where you have more free water, meaning less solutes, to the side with less free water. We'll definitely spend a little bit more time in class talking about this, but basically what happens is water, because it has so polar, it's going to interact with anything that's mixed with it. So if it's not able to mix with anything, then we would say it's free. So only free water, water that's not mixed with anything, will be able to diffuse through a membrane. So the more stuff that's in the water, the less free water there is. So before we move on, we want to review some terms here. So some terms related to a solution. Salt is going to be the solute in a solution. Water is going to be called the solvent. So if I look here, I have multiple solutions, and what you need to do in these is come up with what's the concentration. So the concentration in case of like salt and water, we usually talk about it as being a percentage of salt. So usually it's the concentration of a solution is not by the amount of water in the solution, but instead by the amount of solute in the solution. So the way you do that is you figure out the amount relative to the overall amount. So in this case, what you're doing is I'm adding up, I have 3 salts, and then I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 waters. So that's a total of 21 materials. I think I totally counted that wrong. So 19 total molecules. So 3 out of 19, that gives us 16% salt. This one you can see here is 61% salt. So again, it's the number of salt divided by the total number of molecules. So the other way you could think of this is then, well, if it's 16% salt, that means it's 84% free water. 61% salt, 39% free water. 42% salt, 58% free water. So the higher the solute concentration, the less free water there is. Because there is free water both inside and outside the cell, the direction free water will move will be determined by the relative amount of solute inside compared to outside of the cell. So in the previous slides, I was just showing you, here's the concentration. Now in this one, I'm showing you, here's the cell inside of a watery solution. So inside the cell is going to be its own watery solution called the cytoplasm or cytosol. All right, so the first thing we want to do is figure out what's the concentration of salt inside the cell. So I have 1, 2, 3, 4 salts. So 4, if I use my calculator here, 4 divided by 15 is going to be 27%. So that means 100 minus 27, that's going to give me a concentration of free water of 73%. So what's the solute and free water concentration outside the cell? So how many salt do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 divided by 27 is 33%. So the amount of free water is then going to be 67%. So where is there more free water? Well, there's more free water inside the cell. So if there's more free water inside the cell than outside, then the movement of water will be this way. So you would say the movement of water is going to be out of the cell. So it will, you would say it would lose water. Okay, so again, I did that because I see it has a higher concentration of solute outside than inside, but therefore it's going to have a higher free water concentration inside than outside. So water will move from high free water to low free water. Or you could say it goes from high solute to lower solute. Or sorry, from low solute to high solute. Solutions with equal amounts of solute inside are called isotonic. Iso means equal. Hypo means less, and hyper means more. And tonic, or tonicity, refers to the concentrations of solute. So hypotonic means there's less solute than inside the solution. A hypotonic solution is going to have more solute. These are going to be relative to other solutions, but generally we're going to focus on what's outside the solution compared to inside. So on this, I have these different cells here. So this is 3% salt, so it's 97% water. This is 0% salt. We call that distilled water. So this is going to be 100. So from high to low, water, the distribution of water is going to be that way. We would call this solution outside hypotonic because there's less water. Or sorry, less solute, more water. The outside solution here we're saying is 82% starch. So this would mean that it's going to be 18% water. Over here, 66% starch. That means it's going to be 34%. Again, movement here is going to be out. In this one, 4% and 4%, that means it's 96% and 96%. We would say the movement is going to be both equal in and out. So to define these, this one here is going to be hypotonic outside. This is going to be hypertonic inside. This is going to be isotonic. Again, looking at the concentrations. So this has less solute outside. This has more solute outside. This has equal solutes inside and outside. Okay, let's look at those terms again. These are three major terms to know: hypotonic, hypertonic, isotonic. So this is showing blood cells. And one of the reasons why when you get, if you ever have to get an IV, you're not given an IV of just straight water. You're given an IV that has saline in it. And that's because your body has a certain amount of solutes in it. So your blood must be maintained at a proper solute concentrations. The fluid surrounding your blood is called plasma, and the amount of salt in your plasma must be kept in balance, and it's done by your kidneys. Otherwise, your cells would be affected by osmosis. So if your cells have too much salt outside, they're going to shrivel and look like this. Normal, it should look like this. And if there's not enough salt on the outside, if you get straight water as an IV, this is what's going to happen to your blood. They're going to explode. Note these terms. The shriveling is called plasmolysis. Exploding is called cytolysis. So if we are, let me go back to here for a second. If we are matching these up, we would say this one is going to be your isotonic plasma. Plasma is the liquid outside. This one where it's shriveling up. Where it's shriveling up is going to be the hypertonic environment. Because again, there's more salt outside than inside, and therefore it would shrivel up. And then this is going to be a hypotonic environment. So this is where there's no salt outside, and therefore water would travel into the red blood cells and cause them to explode. So if you're a unicellular organism and you don't have a kidney to help maintain your salt balance, then you need something else. So we talked about this in the last unit. Organelles called contractile vacuoles are necessary for organisms that live in a hypotonic environment like protozoans. They live in a pond. They're constantly taking in water. And basically what happens is the contractile vacuole fills with water and then pumps it out. So this is the filled up with water version of the contractile vacuole. This is what it looks like when it's pumped out the water. Finally, active transport. Most energy from your food is used for that. 60% of the calories you burn in a day are done to do active transport. So active transport requires ATP, and there's generally two types. Molecular active transport, which is often just called active transport. It's where you're going from low concentration to high concentration. Please note that. I have that circled and starred. Then there's bulk transport, and that's where you're forming vesicles. Again, you're going to use ATP, but it's more like a muscular movement than it is where it's using a pump. Molecular active transport involves membrane-bound carrier proteins called pumps, and these pumps work on ATP. And basically what ATP does is it gives up its phosphate group to the pump, and the phosphate is more or less what is like the fuel for it. It kind of is an engine. Every time it gains a phosphate, the pump works. It pumps a certain amount of material through. So here's your ATP, here's ADP. When the phosphate is added to the pump, it then gives it the energy to move the materials against their concentration gradient. So here is a picture of active transport, and that's the one that I'm showing here is called the sodium-potassium pump. And one of the things you might know about your nervous system is that it runs on electricity. This is how your body makes electricity. And again, this is what most of the calories you burn in a day are used to do, is to make the electricity of your body. So it starts with sodium attaching to this pump, and then ATP attaches a phosphate to the pump, and that causes it to pump the sodium out. Potassium comes in, the phosphate group detaches, and then potassium comes in. You don't have to know all that stuff. All you need to know is ATP is used, and it moves materials against the concentration gradient. By moving that, it's going to create a high concentration of sodium and a high concentration of potassium, which is going to lead to it having electrical charge. And that basically, the electrical charge allows you to do things like see, hear, touch, taste, smell, learn, all the things that your nervous system are involved with. So again, key thing, moving against the concentration gradient, using pumps and using ATP to do that. Then you have the other type of active transport called bulk transport. We've actually seen pictures of this already. The term for the bulk transport that we've just called ingestion, it's called endocytosis. Your white blood cells uses antibodies as receptors to identify specific things, and it can then ingest materials that it can recognize. So this is what you want like your body to do with COVID. If it recognizes COVID, because it has the antibodies for it, it's able to ingest those COVID viruses and prevent them from infecting you. And that's, you know, when you get a vaccine, that's basically what they're doing, is they're giving you these little materials here that will cause you to form antibodies. Finally, you have exocytosis. Exocytosis is going to be where those vesicles are going to fuse with the membrane and then release their content. So again, we've illustrated this, we've called it secretion or egestion, but it's also just referred to as, the fancy word for it is exocytosis. So the illustration of it you should be familiar with, just the name now, we're adding a new word, exocytosis. So we've talked about lots of terms here. diffusion, facilitated diffusion, hypotonic, isotonic, hypertonic, osmosis, endocytosis, exocytosis, active transport. These are all key terms that you need to be very familiar with. So if you need to, watch part of the video over again. Make sure you have all the notes filled in before you hand them in and have them ready for your test. All right, so that is the conclusion of these notes.